Hi students, coming to the next class is the translation process, right? See, in the last class we discussed about transcription, and after that we discussed about post-transcriptional modifications. See, in that case we have discussed there is a 5' cap synthesis, right, and 3' poly A tail synthesis, right, and splicing mechanism. And these three concepts we discussed. One is the 5' cap and 3' poly A tail is supporting for the stabilization of mRNA and enhances the lifespan of mRNA. And the splicing mechanism, removal of introns, means coding and non-coding sequences are there, and non-coding sequences are deleted and coding sequences are tagged, right? So I said eukaryotes having introns, and archaebacteria having introns. So based on that, we have to say one statement: post-transcriptional modifications are absent in prokaryotes. Post-transcriptional modifications are absent in prokaryotes. Post-transcriptional modifications are seen in eukaryotes. Right? See what I am telling here, because in prokaryotes introns are absent, right? And we discussed the 5' cap is supporting, for the transportation of mRNA from nucleus to cytoplasm. Nucleus is present in eukaryotes, right? Nucleus is present in eukaryotes, right? And the 3' poly A tail, that one is also supporting for the transportation of mRNA from nucleus to cytoplasm. So what I am telling here, see whatever that mRNA structure is there, this is the mRNA structure. There is a 5', right? This is the 5', and this is 3'. See, 5' side there is a cap structure is there, and 3' side there is a polyadenine, means adenine residues are added, right? So here 5' cap and 3' poly A tail, these structures are especially seen in only eukaryotes, and the introns are only present in introns are only present in eukaryotes generally, but archaebacteria also contains the introns. Right? So here what I am telling, whatever that introns, the cap synthesis and the tail part synthesis, so all these are included in post-transcriptional modifications. So post-transcriptional modifications are seen in eukaryotes. And I like to explain one of the point is here. See, for suppose this is the mRNA. See, for suppose this is the cap region, and this is the tail part or tail region. See, after the cap, and along with the tail part, along with the poly A tail part, UTR regions are present. Within mRNA, there is an UTR regions are present. Untranslated regions. For suppose this is the cap. See, this is the one of the UTR region. And this is the tail part, and this is one of the UTR region. Here AUG codon is present. This is the starting point. So at starting point, the starting codon is the AUG. So what I am telling here, see, AUG is the starting codon, this is the starting codon, then here protein synthesis takes place. Before the AUG codon, some regions are there. This is the termination codon. UAA. This is one of the termination codon. So after the termination codon, some sequence are there. So these regions, especially before the initiation codon, after the termination codon, whatever the regions are there, these regions are called as UTR regions. UTR regions indicates here untranslated regions which are not involved in translation process. The sequences which are not involved in translation process. The sequences, unable to code for amino acids. The sequences are not coding for amino acids, right? So here, this is about that UTR sequence. So UTR sequence sometimes asking, along with that 5' cap and 3' poly A tail structure within mRNA, some sequences are present which are not involved in the protein synthesis process. These sequences are called as UTR, untranslated regions, right? So that is about just introduction regarding to the translation process. Because see, always translation means here protein synthesis. We know the translation is nothing but synthesis of proteins from mRNA. We know trans indicates here changing the information. Here exactly the information is changing from one form to another form. So here information changing from one form to another form. So that one is nothing but trans. We know about the translation. Okay. So here that the same meaning here trans is nothing but changing. The information is changing from one form to another form, right? So that is about that translation process, right? Synthesis of proteins from mRNA. We know synthesis of proteins especially requires the amino acids. We know about the amino acids. See, before discuss about the amino acids, machinery for protein synthesis. See, some of the components are some of the biological components which are present within the cell, which are required for the protein synthesis. Some of the components which are required for the protein synthesis, right? So within that case, the very first one, amino acids are raw materials for the protein synthesis. Amino acids are the raw materials for the protein synthesis. We know here amino acids, according to the NCERT, there is a 20 types of amino acids are present. There is a 20 types of amino acids are present, right? See, generally selenocysteine, that is the 21 amino acid. Generally 21 is there, right? But according to the NCERT, here 20 amino acids are there, and these 20 amino acids, in these 20 amino acids, right, there is a 10 essential amino acids are there and 10 non-essential amino acids are there. We already discussed within biomolecules. See, amino acids here, essential amino acids which are not synthesized in our body. The essential amino acids are not synthesized in our body, which are taken through the diet. The amino acids which are not synthesized in our body, okay, these are taken through the diet. These are amino acids are called as essential amino acids, and 10 non-essential amino acids are there, right? These non-essential means which are synthesized in our body, right? These are called as non-essential amino acids. So we already discussed within the biomolecules chapter, right? So that is about that amino acids, right? So here the very first one, the amino acids as raw materials for the protein synthesis. Amino acids are raw materials for the protein synthesis. We already discussed the proteins are nothing but repeated units of amino acids, right? There is a many number of amino acids are there. Here proteins are heteropolymers. Proteins are heteropolymers. Heteropolymers means here 20 types of amino acids are there. All these amino acids are each type of amino acid having different structure. So here some are there is a side chain containing especially within amino acid we are discussed about. This is one of the carboxyl group is there, right? And this is one of the amino group is there, right? And there is a side chain R is present. There is a side chain R is present. So here R is changing. The R is changing from one amino acid structure to another amino acid structure. So here what I am telling here, in R place some amino acid contains, maybe the glycine, one of the best example, glycine. Here glycine contains the hydrogen atom. Glycine contains the hydrogen atom, right? Maybe other amino acids are there, alanine or valine, leucine. So there is an aliphatic side chain is present. Aliphatic side chain is present. Some amino acids contains the basic groups. Some amino acids contains the acidic groups. Some amino acid contains the aromatic rings. So here different types of amino acids are there. All these amino acids are polymerized with the peptide bond. So the proteins are heteropolymers. The proteins are heteropolymers, right? So here the very first requirement for the protein synthesis, amino acids. Amino acids as raw materials. Amino acids as raw materials, right? So that is about that first point. And coming to the next one is the DNA has the specific control. DNA has the specific control. So here DNA has the specific control means, see, within DNA we already discussed, within DNA also there is an exons are there, there is an introns are there. There is an exons are there, there is an introns are there. Again, my simple explanation from the basic level is, this is the nucleus. Nucleus contains that DNA. Within that DNA, right, there is a tightly packed region is there and loosely packed region is there. The tightly packed region is called as heterochromatin. The tightly packed region is called as heterochromatin. Heterochromatin, okay, not involved in gene expression process. Euchromatin involved in gene expression process. We already know why heterochromatin not involved in gene expression process. There is no space to bind RNA polymerase enzyme. There is no space to bind RNA polymerase enzyme. There is no space to bind for the gene expression process. Means there is no space to bind the components involved in the gene expression process. Simply RNA polymerase enzyme. I am saying only RNA polymerase enzyme. Along with the RNA polymerase enzyme, different components also required. So here, euchromatin, loosely packed region. Okay, see, that is the euchromatin. Euchromatin involved in the gene expression process. There is a tightly packed region is there, that is called as heterochromatin. Okay, that one is called as heterochromatin, right? Heterochromatin not involved in gene expression process. Heterochromatin not involved in gene expression process. So that is the difference between that euchromatin and the heterochromatin. So whatever that loosely packed region is there, within that loosely packed region also, all the DNA fragments are not involved in gene expression process. Here DNA also contains that specific sequences. These are called as cistrons. These are called as cistrons. The functional part of the DNA is called as cistron. The functional part of the DNA is called as cistron. So a bit of DNA, okay, involved in gene expression process. And see, some of the sequences which are not involved in the gene expression process, these are called as introns. So for suppose this is the entire DNA, the red color, right? See, within DNA there is in specific sequences are there. For suppose this is the specific sequence, this is the cistron. The cistron involved in gene expression. For suppose this is another cistron. So many number of cistrons are there. The cistrons are involved in gene expression process. The cistrons are involved in gene expression process, right? So that is about that the DNA has the specific control. DNA has the specific control means the specific sequences of DNA are cistrons. Specific sequences of DNA are cistrons, right, which are involved in gene expression process, which are involved in the gene expression process, right? Because here these are especially involved in gene expression because the specific RNA polymerase enzymes. Why these are involved in gene expression process? Why the cistrons are only involved in gene expression process means, okay, here the cistrons contains there is in particular promoter sequences. There is in particular promoter sequences. The promoter sequences are supporting for the binding of RNA polymerase enzyme. Once RNA polymerase enzyme is binded, there is an mRNA synthesis takes place. mRNA synthesis takes place, then protein synthesis takes place. mRNA synthesis takes place, protein synthesis takes place. So here what I am telling here, remember all of you, why cistrons have the capacity for the gene expression, why introns not have the capacity? Here the simple answer is promoter sequences. The promoter sequences are especially supporting for the binding of RNA polymerase enzyme. Then the particular bit of DNA involved in gene expression process. Then the particular DNA then the particular bit of DNA involved in gene expression process, right? See, so within gene expression process, the regulatory steps of especially in eukaryotic cells. In eukaryotic cells, the regulatory steps for the gene expression is, see, one is the transportation, means one is the transcription. The very first point is transcription. The very first point is the transcription. See, once the transcription takes place, then gene expression is started. Once the transcription takes place, right, mRNA production takes place. mRNA production takes place. Once mRNA production takes place, the mRNA transported after that 5' cap, and 3' poly A tail synthesis, transportation, right? once the transportation is takes place. So transportation also regulating here. If the transportation is not takes place, there is no chance for the gene expression process. So here what I am telling, during gene expression process or within gene expression process, the very first regulatory step is the transcription. The very first regulatory step is the transcription, because once the mRNA production is takes place, these steps are automatically takes place. So this is the very first regulatory step. After that, 5' cap, 3' poly A tail. These are transporting for the transportation. So transportation of mRNA, and last step is the translation. The last step is the translation, right? So these are the major regulatory steps. These are major regulatory steps for gene expression process. So all of you remember here there is an transcription is takes place. The transcription is the main regulatory step. After that, transportation, right? See, between these two there is a 5' cap and 3' poly A tail. Post transcriptional modifications are takes place. After that, transportation is takes place. So after that transportation, right? See, translation is takes place. So here all these are takes place in eukaryotes. All these are takes place in eukaryotes. In prokaryotes, there is no different compartments. In prokaryotes, there is no different compartment. For suppose this is the one of the eukaryotic cell. Within eukaryotic cell, these are the DNA segments, right? These segments are especially involving, especially the DNA segments. These are called as cistrons, which are involved in mRNA production. Once mRNA production is takes place, for suppose this is the one of the mRNA structure. Once mRNA production is takes place in eukaryotic cell, this is the first regulatory step. After that, the mRNA molecules are transported into cytoplasm, from nucleus to cytoplasm. Within cytoplasm, there is a protein synthesis will be takes place. There is a translation will be takes place. That is about that eukaryotes and different compartments. One is nucleus, other one is the cytoplasm, and between these two there is a transport is there. So these are the regulatory steps. But in prokaryotes are not like that. There is no different compartments. This is one of the bacteria cell. This is the DNA. Here very less complexity is there, right? And there is no entrance here. There is no entrance here. Once mRNA production is takes place, suppose this is the DNA involved in the mRNA production. This is the DNA involved in the mRNA production. See, while the transcription is takes place, that time ribosomes are binded. Right? Immediately, see what I am telling here, within prokaryotic cells, same time, there is a chance for transcription and translation. Means, here mRNA production is takes place. When the mRNA production is takes place, that time ribosomes are binded. Immediately ribosomes are binded. See, without finishing here transcription process also, the ribosomes starts the translation. Because, in prokaryotic cells, there is no different compartments. Means DNA replication, transcription and translation. All these takes place, only in cytoplasm. Because there is a, within prokaryotic cells, there is no nucleus. Only cytoplasm is there. Right? So that's the reason why within prokaryotic cells, right, there is a transcription, translation. Both these are occurs same time. Means once the transcription started, immediately ribosomes are bind to the mRNA. Same time, the ribosomes are bind to the mRNA. So within prokaryotic cells, there is an transcription and translation, right? The transcription and translation same time occurs. But in eukaryotic cells, that is about the prokaryotic cells. In eukaryotic cells, the transcription, in one compartment. After that, post-transcriptional modifications. After that, transport. After that, translation. So, in eukaryotic cells, there is a many number of regulatory steps are there for gene expression process. In eukaryotic cells, transcription is the major regulatory step, right? After that, post-transcription, 5' cap, 3' poly A tail, right? After that, transportation is the next step, and last step is the translation. The last step is the translation, right? That is about the regulatory steps. Regulatory steps within eukaryotic cells. In prokaryotic cells, once the mRNA production is started, okay, immediately ribosomes are binded and protein synthesis takes place, right? So that is about the difference between the prokaryotic gene expression regulation and eukaryotic gene expression regulation, right? So here the reason DNA has a specific control. See, this is the overall here. The main point is here. DNA have that specific DNA sequences which are called as cistrons, which are only involving gene expression process, which are only involved in the mRNA synthesis. Why cistrons are only involved means here promoter sequence. The cistrons are contains the promoter sequences. These promoter sequences are recognized by the RNA polymerase enzyme. Then mRNA production. Once mRNA production automatically involved in the protein synthesis. So once the proteins are synthesized, these are maybe the structural proteins, these are maybe the enzymes. These are maybe the enzymes, right? And ribosomes are the protein factories. When you know about the ribosomes from the first year onwards, or maybe 7th class onwards, right? Ribosomes are the protein factories. Ribosomes are the protein factories, right? See how the ribosomes have the capacity to synthesize the proteins. How the ribosomes have the capacity to synthesize the proteins. See, ribosomes have the capacity for the protein synthesis because we already discussed so many number of times, right? Because this is one of the ribosome, right? This is the large subunit of the ribosome, and this is the mRNA structure. This is the mRNA structure. This is the large subunit of the ribosome. This is the 5 prime, and this is 3 prime. This is 3 prime, right? And this is the small subunit of the ribosome. This is a small subunit of the ribosome, right? This is the small subunit of the ribosome. Within ribosomes, different sites are there. The very first site, this is called as A site. The site is called as A site. After that A site, there is a P site is there. This site is called as P site. And the last one is the E site. The last site is the E site. This is E site. Within ribosomes, majorly 3 sites are there. The ribosomes are supporting for the protein synthesis process. And in this case, 3 sites. A site is aminoacyl A site. The very first one is the A site. Aminoacyl tRNA binding site. Aminoacyl tRNA binding site. The very first one is aminoacyl tRNA binding site. Means whatever that loaded tRNA, especially clover leaf structure, tRNA have the clover leaf structure, and the tRNA having different loops are there. This is one of the amino acid binded arm or amino acid acceptor arm. Amino acid acceptor arm. So whatever that loaded tRNAs are there, means if the tRNA binded with amino acid, these are called as loaded tRNAs. The loaded tRNA is also called aminoacyl tRNA. Aminoacyl tRNA. The aminoacyl tRNA binding site. This is the very first site. The binding site is called as A site. A, okay, right? A indicate for aminoacyl tRNA binding site. Right? After that, where the peptide bond formation takes place. Where the peptide bond formation takes place. The site is called as P site. The site is called as P site, right? Last site. Once the amino acid is removed, means once the amino acid is downloaded, once the amino acid is downloaded from tRNA, right? The remaining tRNA, the downloaded tRNA, the amino acid downloaded tRNA enter into the E site. The amino acid downloaded tRNA enter into the E site. So, E site indicates for exit site. E site indicates for exit site. The downloaded tRNA means already here, the tRNA contain amino acid. The tRNA contain amino acid binded here. Okay, the amino acid involved in the peptide bond. So, one of the empty tRNA is present. empty tRNA is present. The empty tRNA enter into the E site. From E site to outside to the ribosome. From E site to outside to the ribosome. Right? So this site is called as exit site. This site is called as exit site. Right? So that is about the A site. And next one, P site. Here P site indicates the site involved in the peptide bond formation. Site involved in the peptide bond formation. Exit site means the empty tRNAs are moving out from ribosome. Moving out from ribosome, right? So the site is called as E site. That is about the ribosomes which are involved in protein synthesis, right? Next one we discussed about the steps in protein synthesis process.